And whenever you're ready, please take it away and tell us about predation, the evolution of bird plumage coloration.
Okay, so all working sound and slides, right? Okay, so thank you so much for being here. Good day to everyone. My name is Ana Leitão. I work at CBU Biopolis in Portugal, and today I'll be talking to you about part of my Marie Curie fellowship, an experiment that I did on predation and the evolution of bird plumage coloration, experimental insights from island and mainland environments. So islands are considered natural laboratories for evolution because oceanic islands are ecologically isolated and relatively simplified ecosystems, typically home to small genetic limited populations and reduced biotic complexity. These unique conditions drive the evolution of distinct traits in island species, often markedly different from their continental counterparts. So among these traits are secondary sexual characteristics like plumage coloration, which can be expressed by both females and males, and that have been shown to decrease on islands. So in this figure here, it's work from Claire Dautrelin and colleagues, and they did a comparative study showing a comparative study with island and mainland species, and they showed that in the number of color patches, the color volume and brightness, it all decreased from mainland to island. Animals are expected to face a trade-off between producing color ornaments and to communicate with conspecifics and avoiding detection by predators. So visually oriented predators, such as raptors, may exploit these conspicuous signals to locate the prey, potentially limiting the evolution of elaborate coloration. Some empirical and comparative studies in a variety of taxa supports this, showing that conspicuous coloration can increase detectability by predators, although there are several studies that also show the opposite. But I will not focusing on it today. In this particular case of islands, in this study here from this figure that I'm showing, it's a study from Bliard and colleagues, and they show that islands have low predation pressure and species facing less predators are more likely to be colorful, even if overall islands support less colorful species. However, experimental tests on the impact of predation on plumage evolution remains underexplored, and especially on island environments. So in this study, we used island and mainland comparisons to experimentally test whether predation pressure differs between island and mainland, and whether this variation is association with differences in color. For this, we used 3D printed bird models to quantify attack rates across island and mainland sites. Specifically, we wanted to answer 3 questions. Does predation differ between island and mainland environments? We predicted that attack rates would be higher on the mainland than on islands, where predation pressure is expected to be higher. We also asked whether coloration conspicuousness relate to higher predation risk. We expected that more colorful and contrasting 3D models would elicit higher attack rates than duller, less colorful ones, because conspicuous coloration can increase detectability. And lastly, we also wanted to know if male-like models suffer higher predation than females. And we predicted that male models would experience higher risk because males typically display more elaborate colors, and so they incur a higher risk of predation. So going to the methods. As I said, we used 3D printed bird models representing adult females and males of different species that inhabit island and mainlands to quantify attack rates across island and mainland sites. The study involved 12 species, 6 pairs of island endemics and their closest relatives. And this is, so from the left, we work out the 3D models and we build this, the ones that are on the slide. So how did we build the models? We used 3D photogrammetry of taxidermic specimens that you can see here on the figures, and these are examples. And we painted the models with colors that replicate the natural plumage from the target species, and this was also based on spectrophotometry. So after achieving close visual matches between the model and the specimen colors, we used avian receptor noise limited visual models to assess color similarity. And this approach confirmed the strong correspondence in chromatic contrast between the 3D models and the bird specimens. So after that, we took the models, and the experimental study took place in several locations where the island and mainland species occurred in the Atlantic Micronesia. So we did in the archipelagos of Madeira, Azores, and the Canary Islands, and Portugal mainland in Europe. So the experiment consisted in setting up several transects per species site, each containing approximately 40 models, half of each model species and each sex. And the transect locations were selected where the target species occurred. We put the models on the ground with a nail and a magnet, and we placed them alternating by species and by sex. And the models stayed, we checked them every 24 hours, and they stay for 48 hours, and then we change site. To confirm the identity of putative predators, we used trail cameras on a sample of models. We also spec the background where we place the models to use in the visual models. And we also characterize the vegetation cover using extracting a normalized difference vegetation index from satellite rasters, but I will not be talking much about it today. And as I said, we use the visual models to compare the colors of the models that we painted, to calculate detectability by visual predators in their natural environments, and to calculate chromaticity and luminance of the models. And the statistical analysis consisted of several general linear mixed models to test whether these lodgement rates, so a proxy of attack rates, differed between island and mainland, controlling for different variables and analyzing the effect of model type and color contrast. So going for the results, these are the main results. We did run several control descriptive analysis and control of the experiments, but I'll be focusing on the main results today. So a total of 3.16% of all models placed were dislodged. This is just a sneak peek of the wildlife that the cameras caught. And although the percentage seems low, this was expected based on other studies on varied taxa. So all good here. So the first question was, do predation rates differ between island and mainland environments? And the answer is yes. We observed an effect of insularity, and we also found an interaction effect of insularity and model color. And this means that we found fewer attacks on islands, the plot on the left, and the plot on the right, we also found that dichromatic models, so the orange ones, were more attacked on mainland than on island. For the second question, is color conspicuousness related to higher predation risk? The answer is yes, but only on brown backgrounds. So it's the first plot on the left. We found an interaction of insularity and chromatic contrast, and a marginal effect, only chromatic contrast. And the right plot, it's against green background. The tendencies are the same, but we didn't find any significant results. So what does this interaction mean? For mainland sites, as chromatic contrast against brown backgrounds increased, the probability of dislodgement also increased, suggesting that more conspicuous models were more likely to be attacked. While on islands, this pattern was not observed, and actually the tendency seemed the opposite. And the third question, do male models suffer higher predation than females? And the answer is no. We did not find a relation between the probability of being dislodged and sex-related colors. So going to the conclusions. So these are the key results. Island models had lower dislodgement rates. Dichromatic models were more attacked on mainland. On mainland, we found that more contrast was related with more attacks, and we found no significant sex-specific predation differences. However, there are some limitations to this work that I wanted to address quickly. Models lack movement, behavioral cues, which may have underestimated predation pressure. And the predators captured on camera were mostly invasive or generalist species. So this means that non-native species may be altering this predation dynamic. So the key takeaways is that we found empirical support for relaxed predation pressure on islands. And so islands may allow ornamentation to evolve under relaxed natural selection. Coloration is more risky on mainland. More conspicuous coloration leads to higher predation, but on mainland. Low attack rates are still informative. So we consider that 3D models can be a useful proxy. And we also believe that this island-mainland comparison is a powerful tool in evolutionary ecology, and we hope we see this sort of work more regularly. And that's it. I think I'm on time. I'm happy with that. If you'd like to learn more about our team, ongoing projects, collaboration opportunities, or student openings, please visit our website and feel free to contact us. And I'm open to questions. Thank you.
Great. Thank you for that really interesting talk. We have a question. Someone raised their hand. Do you want to go ahead and ask your question?
Yeah, hi. Thank you for the talk. Can you go back to the slide where you discussed the third question, if males are attacked more or not?
The results? This one?
Results, yes. Yes. Okay. So model sex diversity. Okay, so in your sample, some birds were sexually dimorphic, whereas some birds were not. So for this comparison, did you treat them differently or not?
Yeah, so on this, good question. So on this model, I didn't give the details, but on this model, we excluded all the monochromatic model species. So only the dichromatic species are here.
Okay.
Okay.
Are there other questions?
I cannot see the chat now, so I don't know if there's any questions.
I haven't seen anything posted in there, but people can post them in the Q&A. I actually have a question.
Okay.
So you have those camera traps. Did you see any differences in predators between mainland and island?
Yes, but in relation to the expected local species.
Okay.
Also, we had to exclude a few transects because of the data that we collected from the camera traps. So, for example, in some islands like Fuerteventura and the Canary Islands, there's a lot of introduced goats, and that's a clear difference. And also introduce squirrels and such. And so for this reason, the camera traps helped to be sure that we were collecting reliable data. So in all these cases, we excluded the transects from the analysis when we saw that there was like a herd of animals.
Yeah, very interesting work. Are there any other questions? Oh, we have something in the Q&A. There's a question here that asks, do your models mimic the species in size? Are the species variable in size?
Yes, that's a good question. Yes, they do mimic. So because we did the photogrammetry of the real specimens, we use the textures and the size of the species. So yeah, they mimic size.
Great. Well, I think with that, we will move on to our next speaker. So thank you so much. And our next speaker is Tuhin. Do you want to share your slides?
Yes. Can you see this?
Yep, I can see them fine. So you may go
Okay.
ahead and get started whenever you're ready.
Okay. So, hello everyone. My name is Tuhin Subrupal. I am currently working as a senior research fellow under supervision of Professor Anindita Bhadra in ISA Kolkata, India. Today, I'll be talking about uh, how adult and juvenile free-ranging dogs navigate aversive foraging challenges differently. Now, is it changing or? Okay. So, we are currently working on Indian free-ranging dogs. They are basically ownerless canines whose movements and reproductive activities are not controlled and confined by humans. Also, they evolved from grey wolves 15,000 years ago, and among the whole population of the dogs, 70 to 80% are considered as free-ranging. Now, I'm working on the feeding behavior of free-ranging dogs. Now, feeding behavior is shaped by the early experiences or trial and error procedures. So, in case of the juveniles, in different species like primates and also the some bird species, juveniles foraging behavior and also their dietary preferences differs from adult ones. Now, juvenile learns through various exposures like trial and error and different positive and negative exposures. And by the trial and error and different exposures, they fine tune their complex skills. Now, in case of the free-ranging dogs, we conduct several experiments on the adults and also on the juveniles. Now, what is the scenario in case of the juvenile free-ranging dogs? How does the behavioral reaction of juvenile free-ranging dogs vary from the adult free-ranging dogs? That is our main query here. Now, as we all know, that free-ranging dogs are primarily scavengers. They live on human-generated waste, and they often scavenge from the garbages or trash cans. They have a preference on protein-rich foods, and they follow a rule of thumb. That is, if it smells like meat, get it or eat it. They follow the rule of thumb to maximize their protein intake. Now, in Indian context, we often use lemon, citrus lemon, in our Indian diet. So after using, we often throw it in the garbages. So while scavenge from the garbages, free-ranging dogs often encounter lemon and the lemony items or the lemon parts along with their palatable or preferable foods. Now they can't avoid it, but in case of the pet dogs, uh, they mostly rely on their owners for their foods. I mean, owners provide them foods, so they carefully choose the type of the food that has to be provided. But in case of the free-ranging dogs, they survive by scavenging. So they have to encounter these avoided food items. So my previous research, so one paper has been published in this year only in applied animal behavioral ecology, applied animal behavior. Adult ranging dogs showed avoidance towards lemon, the whole lemon, and also in among the three parts of the lemon, that is the lemon pulp, lemon cell, and the lemon juice. They avoid lemon juice compared to the other items. Now, we provided three different concentrations of the lemon juice: 50%, 33.3%, and the 25%. The above 50% is really toxic for them. So among these three different concentrations, adult free-ranging dogs prefer to scavenge from the lower concentrations, that is the 25% lemon juice concentration, compared to the highest one, that is the 50%. But in case of the juvenile free-ranging dogs, what is the scenario? That is my question that I am going to explain. Now, to explore that particular area, I designed a simple experiment where I provided a biodegradable bowl, and in the biodegradable bowl, there are 20 ml of solution along with a 15 grams of chicken. So basically, most palatable food and least palatable solution has been mixed. Now, I provided three different conditions, that is 50% lemon juice, 33.3% lemon juice, and 25% lemon juice. Others are constant, like the amount of the solution and also the amount of the chicken is constant. And on a particular cardboard of approximately 1 meter, we placed the three bowl 0.3 meter apart, and we placed in front of the dog and waited for 3 minutes for their reaction. We recorded in mobile camera their reaction, and we after the experiment we decoded the videos for them. So we conducted a total of we conducted our experiment on total of 94 juvenile free-ranging dogs, and out of them we have taken 73 data out the experiment video, and we conducted our experiment in three different time slots, that is 6 to 9 a.m., 9 a.m. to 12 p.m., and 3 p.m. to 6 p.m. Now, to reduce biases, we choose isolated dogs mostly, and if that is not possible, we isolate a particular dog from a group to conduct our experiment. And also, we haven't provided any pointing cue during the experiment to reduce the biases. Now, we conducted the experiment in 14 different places in and around Kalyani of West Bengal, that is situated in India. Now, that is an experiment video where a juvenile dog is choosing from one of the three bowls. Now, coming to the results. After decoding, we conducted statistical test to get our results. So there we have checked the frequency of the events. So we checked the sniffing and licking behaviors. There was no differences. In case of the strategy making or strategizing events and the consumption events, that is the eating behaviors, juveniles show consistent behaviors across all concentrations. But adult dogs do discriminate. Adult dogs so strategy making or strategizing behavior. So strategy making behavior is an umbrella term. There are 16 different behaviors they show before going into the eating behaviors. So basically sniffing, licking, then strategy making, and then going to eat a particular food item. So in case of the strategizing and eating behavior, adults discriminate and they prefer to show these behaviors in case of 25% bowl compared to the highest one, that is the 50%. So we conducted a generalized linear mixed modeling where we have seen that adults had significantly higher likelihood of engaging into eating behaviors than the juvenile ones. And also higher lemon juice concentration deterred female dogs likelihood of eating behaviors than the male ones or the male free-ranging dogs. In case of the strategizing behavior or strategy making behavior, adults are likely to engage in strategizing behavior more than the juvenile one. And also the 50% concentration is associated with the reduced likelihood of the strategizing behavior or strategy making behavior compared to the 25% or the lowest concentration of the lemon juice that has been provided to them. Now also we have conducted the Markov chain to understand the transition probabilities of these different okay, so it is not changing. Can you see the Markov chain slide?
No, it appears stuck on this slide.
Can you hear me?
Yes, now we can. Yeah,
Really sorry.
it's okay. You can share your slides again.
Can you see?
Yes, we can.
Oh, it is okay. So yeah, so we conducted the Markov chain analysis to understand the probability of the transition from one behavior to the another. So before my experiment, previous experiments showed that the chain of events were sniffing, licking, and eating. So my experiment showed that that is not the case. So there is an intermediate behavior. There is an umbrella term. Basically, there are 16 different behaviors that I have explored. So the chain is basically sniffing, licking, strategizing, and then eating. You can see the probability of the events here. That is constant for juvenile dogs and the adult dogs for each concentration, 33, 50, and 25. So hence it is proved, or it is being shown, that the strategy making event is an important factor that linked leaking behaviors and eating behaviors, working as a bridge. So we formed a Sankey diagram that is basically a sequence pattern or sequential flow of events. We can see the progression of the progression of the dogs from initial action to the final outcome. That is sniffing, strategizing, licking, or licking, strategizing, and then coming to the eating. That is for the adults. And the numbers are the actual numbers of the dogs we conducted our experiments. And for the juveniles, compared to the adults, it is difficult to find the juvenile dogs. So the number of the juvenile dogs is less compared to the adults. Now, this strategizing behavior or strategy making behavior, is it that important? So here we have find out that the eating after strategizing, that is significantly higher compared to the eating without strategizing or strategizing but no eating. So yeah, we can say that for the adult and juvenile both, strategizing before eating is a significant bridge, we can say. Now, what is the final takeaway from this particular setup or particular experiment? Previously conducted on adults, now we conducted on the juveniles, and we are comparing. So here we have seen that juveniles scavenge indiscriminately. They don't discriminate among these three concentrations, or we can say they don't have the skill set to discriminate. But in case of the adults, they discriminate and they scavenge from the lowest acidity, that is the 25% lemon juice, compared to the highest amount. So also we have checked the pH of the solutions also. Here also one more thing, that is male free-ranging dogs tolerated higher concentration of the lemon juice better than female free-ranging dogs. So there we have seen a sex-based differences are there. Also, lemon juice, especially the highest concentration among the three, that is 50%, significantly deterred the strategizing behavior and also the eating behavior compared to the lowest one. So basically 33.3% is not significant compared to both of them. Now, juveniles lacks experiences and skill set. So after the trial and error, they refine their skill set. So thanks to my professor, Dr. Anindita Bhadra and Sagarika Biswas for helping me in the presentation and also the research that is currently in the review process. We have submitted the manuscript, and thanks to the Build Lab for sharing their knowledge. Thanking you for your patience. Thank you for listening.
Okay, Great. Thank you
Any
so
question?
much. I don't think we have time for questions due to the technical
Oh,
difficulties, but if
really
some,
sorry.
it's okay. If the next presenter, Anasawara,
Hello.
hello, do you
Yes.
want to share your slides?
Yeah, sure.
And if there are questions, you can post them in, people can post them in the Q&A and we can try to answer them later, or maybe we will have time at the end. Are you able to share your slides?
Uh, uh, can you see?
We're not seeing any slides.
I'll try once again.
Okay. Yes,
Can you
now
see
we
it
can.
now? Yeah.
Yes. All right. Please.
All right. Yeah. Let me start. Yeah. Hello, everyone. I'm Anasawara from ICER, India. Thank you all for attending my talk. I'll be talking about, So I'll be talking about a study we have done on butterfly species to understand the preference performance hypothesis in butterflies. So let us start with the life cycle of a butterfly. We all might know still, eggs laid by ovipositing females will hatch to become caterpillar or larvae who will eat and one day will decide to become a chrysalis or pupa, which will then eclose to become the beautiful adult butterflies. So from understanding the life cycle, let us now understand the importance of the behavior of egg laying or oviposition. For which, let's imagine a scenario where an ovipositing female is laying her eggs on a concrete wall. What will be the fate of the comparatively less mobile, newly hatched larvae? They'll die. Why? Because there was no food available for them. Now let's imagine another scenario where an ovipositing female is laying her eggs on some random plant in the garden. So what will be the fate of the hatched larvae then? Most of them will die, but some might survive. Now, when are they surviving? They will survive if laid on specific plants on which they can complete their life cycle, or they are termed as host plants. So the decision made by an ovipositing female will directly influence the fate or survival or fitness of the offspring or the larvae. So we can say oviposition behavior is very important. In general, butterflies can have, say, a single butterfly species can have multiple host plants. For example, this particular butterfly is Zizula hylax, which has host plants belonging to multiple plant families. But on the other hand, there are butterflies like Sirocroa thais, which has one or very few host plant species belonging to only one plant family. Now, I already said butterflies can have multiple host plants. But when I said that, it doesn't mean they'll use the plants equally. Rather, there will be a preference hierarchy. For instance, let's imagine if a butterfly species has 4 host plant species, as shown here, red, orange, yellow, and green. They can have a preference or a preference towards only one particular host plants, or preferring most or using most. When I say host use, I already mentioned the term host use. How are butterflies using a host plant? Females or the adult mated females, when ovipositing, when laying eggs, they use the host plant, and also larvae to eat or feed on different parts of a host plant. This is how butterfly species will use its host plant at different life stages. So keeping all this information in mind, one can expect that um, the host plant on which number of eggs laid, most number of eggs laid, and the host plant on which the larvae will survive better should be the same. And this is oviposition preference or oviposition preference larval performance or mother's knows best hypothesis, which states that or which suggests that the mothers give their offspring a better start or best start by laying her eggs on a suitable host plant, host substrate actually. So we tested this hypothesis on a nymphalid polyphagous butterfly, Acra terpsichore, which has host plant belonging to different host plant belonging to multiple plant families. We tested this hypothesis by measuring larval survival for larval performance and oviposition preference or the egg laying behavior. And for larval survival, we provided newly hatched larvae of different host plant. Hello? Yeah. I think I'm audible. So I was at larval survival. So larval survival, we were providing the larvae, newly hatched larvae with different host plants, and we checked for its survival. So if a larvae completes the life stage and pupates, we'll call it to survive. And for the oviposition preference, the ovipositing females were released into a cage with the selected host plants, and we checked for number of eggs laid on which plant, etc. Yeah, we we conducted these experiments on 2 different populations collected from southern India. The first one, the local population from Trivandrum, Kerala, and the population was feeding primarily on Passiflora foetida, the plant Passiflora foetida, and the second population from another region, Tirupur, where the population of Acra terpsichore, the butterfly, was feeding on another host plant, Turnera subulata. Now, we predicted that both the populations will support preference performance hypothesis. Let's see the results. First, oviposition preference or the egg laying behavior. Yeah, here in the y-axis, it's the mean of proportion of number of eggs laid by each female tested, the 35 females, on each host plants. The host plants are given here in the y-axis. So we could see that females chose or preferred 2 host plants equally, Passiflora foetida and Turnera almifolia. On the other hand, larval survival, let me say the y-axis here is a survival percentage. Survival as in larval survival percentage, and on the x-axis, it's the same. It's the host plants. And the larvae here survived better on these 2 host plants almost equally, which are Passiflora foetida and Adenia hondala. So we can see there is a preference performance mismatch in a sense. The host plants preferred by females are not always the best host plant for survival, like Turnera almifolia, where we could see none of the larvae survived. And also host plants on which the larvae survived better, the females did not choose that. Now, for the second population, Tirupur population, the females, the axis of the graphs are the similar as the previous slide. Yeah. So for this population, females preferred Turnera almifolia most, and the larvae survived better on this same particular host plant, Turnera almifolia. So we can say relatively better preference performance match. But yes, larvae has survived on plants which were not selected by the females, like this red one, Adenia hondala as well. Yeah. When I introduced this hypothesis, I did mention that female mothers should choose not just suitable host plant, but also suitable subplant substrate for oviposition. Just imagine if the eggs were laid on a bark of a tree, it is as similar as concrete wall. So let's say substrate is also very important, like flowers. From observations from the wild, we know that larvae of Acra terpsichore feeds on flowers of Turnera subulata and Turnera almifolia. And keeping this in mind, we tested this preference performance hypothesis with flower keeping as a substrate on the second population, Tirupur population, on Turnera subulata and almifolia. And we predicted that females will choose and larvae will perform better when flowers will be provided along with the leaf. And methods were similar to the previous set of experiments, but here for the oviposition preference trials, the ovipositing females were released into a cage with 4 choices: with and without the first plant, with and without the second plant. And these are the results. The mated females preferred host plant without flower and Turnera almifolia without flower. And the larvae survived better when no flowers were given along with the leaves. So they preferred and performed better without flowers. So to summarize the complete talk, what we could understand till now is that Acrea topsicore shows populational level differences in host use. And for the first population, Trivandrum, the local population, we could see a preference performance mismatch. Now, for the second population, we could see that there was a better match. And also, additionally, we could see for this particular population, they preferred and performed better without flowers. With this, I would like to thank Evolution 2025 for the platform, my PI Ulasa Kodadaramaya, and my colleagues of Vanasiri Lab. Thank you.
All right, thank you for that excellent talk.
Thank you.
We do have a question. Anamitra?
In chat, is it?
No, someone's
No,
going to
I
unmute
will ask
themselves. Yeah.
Yeah, sure.
Yeah, hi. Nice talk.
Hi.
What is
Thank
the what is your primary suspect? Why do you suspect this difference between the two populations happening? Same species, but in two species. First of all, the different preference, and as well as the larva survivability. Any
Yeah.
particular reason that might be?
Yes, there are several reasons I can suggest from the literatures and from my observation. One most prevalent one is local abundance of the host plant. So the first population which we selected is the southernmost part, that's the Trivandrum one. So there, Passiflora fetida, on which we have already collected the population from, that is the most abundant among the other selected host plants or the host plants which I have tested. So one primary reason I'll say host plant, local abundance of the host plant. Yeah, that is a one reason, and there are other reasons as well. Say in the course of evolution, they are choosing one particular host plant, and then they start to use it in a different population, and then they continue it. So that's a long-term process. The main reason I would say local host plant abundance. Yeah.
Are there other questions? Apologies if I missed this, but are any of these plants native versus introduced, or are they all native?
Yes, very good question. Actually, I didn't, time constraint, I didn't include that. Among these, one particular host plant which had red colored bar, that is the only one. Adenia hondala, it's the only one native plant to the Indian subcontinent. And this particular butterfly is actually known to be native one. So we think that was using, in the previous time, it was using only that particular host plant. And in the course of time, when the other plants were introduced, they might have thought or they understood the other plants were better, say, yeah, and then they shifted. So yes, in one particular plant on which I am working, one plant is native. Yeah, yes.
Thank you.
Yeah.
That's very interesting.
And to add, the native plants are actually very less abundant right now. So maybe that's a major reason why they chose the introduced ones. Yeah.
Yeah, that's sounds like a great system. All right, I
Yes.
think we'll move on. Thank you so much. We'll move on to our next
Thank
talk.
you. Thank you.
Yeah, so
I think that is me.
Yep, that's you. So if you want to share your slides.
Okay,
Do
we'll
we?
replace the it's fine. Yep.
All right. Our next speaker is Anamitra Roy, and he'll tell us yellow is better than food.
So good morning, good evening, good afternoon, based on where you are. I'm Anamitra, and I will talk about India's dogs and their favorite color. Spoiler alert, it is yellow. So I see many people have joined after Tuhin's talk, so I will introduce the FRD system a little bit. Then I will jump straight into the actual experiments that we did. And after everything is done, I will come back to color preference in light of evolution and discuss why this might be happening. So these are the beautiful dogs that we work with. They are also called the Indian pariah dog. Their movement and reproduction are not controlled by human. They live on the streets, and they have been doing so for many, many generations for hundreds of years. And they have complex group dynamics and territorial behavior as well. And they interact with humans quite frequently. In fact, they are the top scavengers of human-generated food waste. And that is their primary source for nutrition as well. Now, this is basically my experiment setup. 3 terracotta bowls painted in particular colors on a cardboard platform. Why these 3 colors? Because dogs have the ancestral mammal colors cone cell system, which is blue and yellow. And whatever is neither blue nor yellow, they will be seen as shades of gray. If it is bright, it will be white. If it is dark, it will be black. Otherwise, somewhere in the middle, it will be gray. So I chose the 3 hues that the dog can see. And then we went and located solitary dogs in the natural habitat, which is the urban and semi-urban streets of our institute, near our institute, not inside the institute. Then we approach the dog, call the dog, place the setup approximately 1 to 1.5 meter away, and allow the dog to approach. We step back and assume a neutral push posture without pointing gestures or anything, so that whatever choice the dog makes is its own preference. And we allow the dog maximum of 1 minute. Each dog is tested only once so that we can get a population level idea of the color preference, and so that there is no effect of learning affecting consequent choices. And whichever object the dog investigates first, that will be the dog's choice. I have a little video for you guys now. As you can see, a dog on the streets. This is our field. And the dog approaches, in this case, yellow. So what is the result of this? As my title said, these dogs prefer yellow. And this preference is true for both adults and pups, suggesting that this preference might be innate, or if it is not innate, it is learned very, very early in their life. Because we tested pups that were still breastfeeding from their mother, and some post-weaning pups. So if it is either innate color preference or it is learned very, very early. What about blue and gray? There is no difference between blue and gray. Yellow is preferred more than them both, and blue and gray are roughly equal. Now comes the most interesting part of the experiments, strength of preference. Okay, dogs like yellow, so what? Is this preference strong enough to affect their lives, affect their daily lives? Can this affect their foraging? To test that, I gave them a choice of an empty yellow bowl without any food reward and a gray bowl that had some food. The food in one condition was biscuit, in another condition was chicken. And the dog has to understand that there is food in one and no food in the other. Inside the bowl, the food can get hidden. So what we did, we turned the bowls upside down. Now the bowls are working as pedestals. And on top of this gray bowl, now we put the food. So the dog can see the food. Dogs are excellent at smelling. They definitely know the food should be there. Now we are saying, are they going for the food or are they going for the favorite color? Now to establish that they can indeed see the food, the little piece of biscuit that we had used, we did one control experiment where both of the bowls were yellow, but one of them had food. Food was biscuit in this case. So when both of the bowls are yellow, and only one has food, the dogs are going for the biscuit. They can see the biscuit just fine. They like eating the biscuit. They go for the biscuit. But if I just change the color of this bowl from yellow to gray, the whole trend reverses. Now they are ignoring the biscuit and going for yellow. Empty yellow bowl that has nothing, that has not been touched with a hand that touched food, so there is no smell cue. And more interestingly, this same preference stays just like this, even when the food is chicken, and a much bigger piece of chicken than the biscuit, a 10-gram piece of chicken, the same preference persists. So this preference is quite strong. It can affect their daily lives. Two interesting videos for you for this experiment. See, even from this image, you can see, I hope, that the gray bowl has this chicken. The yellow bowl does not have anything, but the dog is already fixated on the yellow bowl. And when it is allowed to approach, it approaches the yellow bowl. This one particular video I find very funny. Approaches yellow first, investigates, no regard to any other option. But then the dog does not go to the gray, goes to the human and starts begging for food. This is not common. Most of the other dogs went for the gray bowl, but this particular dog did begging from the experimenter. Then went back to yellow, and only after that went to the chicken bowl and got the chicken finally. Such strong his preference is. Now the biggest question, why? And the honest answer is that I do not know. Color preference has been observed in many, many different animals, and there is no clear consensus on why this happens. There are theories, of course. One is the species confidence hypothesis that was proposed after working on zebra finches. This basically says that whatever color is on one particular animal's body, for example, the zebra finch's beak is orange, the cheek is yellow, they will have a preference for those particular colors in mate choice context, as well as while in foraging or just novel object choice experiments. However, this particular theory has had some problems with reproducibility. Many studies have failed to replicate the results. So it hangs in the, it has still a big question mark on it. But many species have been seen, in many species it has been seen that the body color does affect sexual selection. For example, in blue-footed boobies, the bluer the feet, the more chance, the more success of reproduction will be seen. There is another theory called the ecological variance theory that says the most usual, the most common value of a color will reflect in that color's preferentiality, will dictate whether I like the color or not. For example, if I see good things that are yellow, I will like yellow. If I see bad things that are blue, I will hate blue. We Indians eat a lot of turmeric, and most of the food that the dogs get from us are yellow. So this could be one possible thing. However, even pups showed the same preference, and they are not yet eating human origin food. They are still nursing from their mother. So I do not think this is a likely explanation either. Humphrey's experiments, he worked with monkeys and saw general interest in some particular colored object or pleasure, which is just a neurological response without any clear reason why this is happening, can dictate the preference. That can also be true. We won't know unless we scan their brains. Just color psychology is a big thing. Some colors elicit emotions, different kinds of emotions, and that might be what is dictating this preference here. So if you guys have any idea on why this might be happening, I am absolutely welcome to suggestions. Anyway, the research so far has been published. And if you want to ask more about this, I am reachable on these 2 emails. I think I have time, so I will slowly move on to my acknowledgements now. Yeah, first of all, the model system. They're very good boys and girls. And thanks to my all co-workers and my professor, Professor Anamitra Roy. So that is it from me. I am now open to questions.
Thank you. Yeah, we have time for questions if anyone wants to post them in the Q&A or raise their hand. Oh, there is a question, several questions
I see them.
in the chat. So the first one is there anything dangerous they learn to avoid that is gray or seen as gray by them? Traps or something like that?
No, these dogs are not really trapped. When people do want to kill them, they just go and beat them up or poison them, mix poison in their regular food and thus poison them. Yeah, unfortunate, but happens quite a bit. People are not really open to free-ranging dogs. But no, nothing as such great that is guiding this. Actually, I have something more to say, but I will move on for now. If you want, I have many stories regarding this. You reach out to me via email if you want to. I will answer the next one now then. Is there any food under the bowl? Can the dogs smell that? So I will go back to my presentation for a bit. In this particular experiment, I hope my screen is visible. Yes. In this particular experiment, the food is inside the bowl. And that is only one condition. I did two conditions with this same setup. One was food inside all these 3 bowls, and another with no food in any of them, just treating the bowls as novel objects. In both cases, I am not showing the no food data right now, but the both cases, the preference is very similar. So presence of food does not matter. And in this condition, in this experiment, there is no food under the bowl at all. This bowl, the yellow bowl has not even been touched with a bowl that touched biscuit or chicken. Completely separate bowls, touched, handled separately. And the food, the only food here is on top of this bowl. So that is completely visible to the dogs.
And the other part of that question was, does the order matter, like left or right? Did you randomize that?
Left or right does not matter. Not really. We did randomize, so there is no consistency there. So there is no association with side and color, basically. Overall, they seem to have a left side preference across many experiments, but like it is not that some color is being particularly chosen when it's on the left or something like that.
All right.
I think one question is in chat. Do I have time?
Yeah, you have a minute or so. Go ahead.
Okay, Franco is asking the dichromatic fission of dogs cannot be an answer or something in related with. I'm not sure I understand the question, but I will try to answer. I chose the 3 particular colors, yellow, blue, and gray, because those are the hues, the pure colors that the dogs can see. So if you take, for example, red, the dogs will see red as kind of a muted yellow. Same thing for green. Green will be seen as a muted yellow thing as well, unless the green is blue adjacent, in which case it will appear gray first, and the closer you go to blue, it will appear more and more blue. So I hope that answers the question.
Thank you so much. I think we'll move on to get our next speaker set up. So Kate, if you want to share your slides.
All right, is that showing up?
We're seeing your notes, not the slides.
Pardon me. Mm. Perhaps can make this full screen.
All right, yes, that looks
Perhaps
great.
not ideal, but it'll work.
Okay.
Cool.
All right, tell us about how territoriality shapes coevolution of cooperative breeding and female song.
Great, thank you so much. Yeah, so good morning. My name is Kate Snyder, and I'm a postdoc at Vanderbilt University. And today I'll be talking about how territoriality shapes the coevolution of cooperative breeding and female song in songbirds. This work represents a collaboration between my undergraduate researcher, Elena Lobren-Pierce, and my advisor, Nicole Crianza. So birdsong has traditionally been studied through the lens of sexual selection, with most research focusing on male song functions like mate attraction and territory defense. As Karen Odom and colleagues showed in 2014, though, female song is actually widespread across the songbird phylogeny and is now considered to be the ancestral state of songbirds. However, the social and ecological contexts that promote female song evolution overall remain pretty poorly understood. We know from various species that female song can serve functions like mate attraction, female-female competition, and territory defense, as well as pair bond maintenance, sometimes involving duets, and gregarious song in European starlings. Stemming from these last two examples, where song is used to promote existing social bonds instead of during agonistic interactions, we sought to investigate whether female song could have pro-social functions on a broader scale. So we did this by focusing on cooperative breeding. However, there's a critical confounding variable that we need to account for, and that is territoriality. Both female song and cooperative breeding are overrepresented in territorial species, and a prevailing hypothesis is that female song largely evolves to assist mates with territory defense. So this leads to three hypotheses for how cooperative breeding and female song might co-evolve. First, if female song serves a primarily territorial function, any link with cooperative breeding would disappear when controlling for territoriality. Second, sexual selection. If reproductive skew increases female-female competition in cooperative systems and female song functions primarily in intersexual agonistic interactions, we might expect more female song to evolve in these systems. Third, pro-social functions to enhance direct benefits of helping. So if song can facilitate group coordination and cohesion, it could be especially beneficial in cooperative systems. So to test these hypotheses, we compiled species-level data on cooperative breeding for over 4,000 Aussian species, female song presence or absence for over 1,000 species, and territorial behavior classifications as well. So we used phylogenetic comparative analyses with a consensus phylogeny from bird tree to account for the evolutionary relationships. So here's our phylogenetic data set showing the distribution of cooperative breeding and female song across 1,041 species that had data for both traits. So notably, the states of both traits are well distributed across the phylogeny. So by raw species counts, there does appear to be an overrepresentation of female song in cooperative breeding species, 81% versus 61% in non-cooperative species. So to test whether this association is evolutionary, evolutionarily meaningful, we used a method adapted from Kielsenbeck that generates stochastic character maps from real data and compares them to simulations that assume trait independence. Essentially, we're asking if these traits evolved independently, how often would we see the overlap that we observe? So this method generates distributions of overlap statistics across many simulated stochastic character maps. So if the real data shows more overlap than the null distribution, we can reject independence. And indeed, female song and cooperative breeding co-occur significantly more than expected by chance, with a p-value of 0.01. The red asterisk shows that cooperative breeding with female song present occupies more of the evolutionary tree than expected under independence. But we wanted to go further and ask about evolutionary dynamics. We generated many stochastic character maps to quantify whether evolutionary transitions in one trait coincided more frequently than expected with specific states of the other trait. And the results reveal some interesting patterns. Most notably, we see far fewer transitions from cooperative breeding with female song to cooperative breeding without female song than expected, shown by that bottom blue arrow. This suggests that cooperative breeding may protect against female song loss. The red arrows show that there are more transitions away from non-cooperative breeding with female song absent than expected. So that is a more unstable state than we would expect. So to incorporate territorial behavior, we used Tobias and colleagues' three-level territoriality classification. This classification is somewhat based on the duration and seasonality of territory maintenance, but we were a little more interested in the intensity of territory defense. And since that middle level of territoriality mixes both strongly but seasonally territorial species, as well as year-round species that are like more weakly territorial, we created new binary classifications to capture both the strength and the exclusivity of territorial behavior. So in this second column, it is our definition of our weak versus strong territoriality classification. And in the right column is our how permissive or exclusive a species is in maintaining a territory. And we define that as a single breeding pair defending an exclusive territory. So that allowed us to kind of capture some of the variation, including species that maybe are colonial nesters or also cooperative. And so this diagram shows how species were reclassified between the original three-level classification and our new binary classifications along these meaningful biological variation in territorial strategies. So the first additional analysis we did here was phylopath. And so these were the four traits that we included in our phylopath model. And these are the relationships that are, that there's at least some hypothesized or evidenced relationships that are already exist in the literature. And this is what we're trying to figure out. Is there an association between female song and cooperative breeding? And if so, is there directionality? And so we find that the averaged top models show consistent support for an evolutionary interaction between female song and cooperative breeding. Importantly, this interaction persists even when we control for territoriality, suggesting it's not just an artifact of their shared association with territorial behavior. So we then again did use the stochastic character mapping to examine transition dynamics within different territorial contexts. And so we find here that within permissive territoriality lineages, we see that non-cooperative breeding with female song present is, again, an unstable evolutionary state, shown by the red arrows, indicating more transitions away from that combination than expected. And this suggests that when territorial pressure is relaxed, female song is either lost or cooperative breeding is gained. And yeah, that's kind of the state we're looking at there. So similarly, in weak territoriality lineages, non-cooperative breeding with female song is unstable. But crucially, in strong territoriality lineages, we see that cooperative breeding protects against female song loss, shown by the blue arrow, indicating fewer transitions from cooperative breeding with female song to cooperative breeding without female song. We followed this up by using phylo-GLM, which allows us to estimate effect sizes. And so these plots show that the effect of cooperative breeding on female song probability is strongest in the weak and permissive territorial contexts, which supports some of our hypothesis that territoriality adds an extra dimension to understanding how female song and cooperative breeding are kind of co-evolving. So, in conclusion, territoriality shapes cooperative breeding in several ways. First, shapes cooperative breeding and female song coevolution. So first, cooperative breeding and female song are linked beyond their shared association with territoriality. Second, while female song is broadly associated with stronger territoriality, cooperative breeding provides additional protection against female song loss in that context. Third, these results seem robust as we controlled for geographic and taxonomic sampling biases through systematic downsampling. I didn't have time to show here. And finally, these patterns indicate some support that the hypothesis that female song serves potentially pro-social functions in cooperative systems, facilitating group coordination and cohesion, rather than just territorial defense. And especially when we take that in conjunction with some other results that I didn't have time to show, but you can find in the preprint that includes some of these results on bioRxiv.
All right.
And yeah, there we go. Yes, I'd like to thank the members of the Koreans Lab and as well as our funding sources from Vanderbilt Evolutionary Studies Initiative and NSF. And thank you, and I'm happy to take any questions.
We have time for a question or two. If anyone, there's something in the Q&A. There's a question about the systematic downsampling, if you could briefly discuss what you did.
Yeah, so so since there is a kind of known overrepresentation of, or like Hall Arctic species tend to be more studied than tropical species. So we did find that that bias is present in our data set. And so we did two things. We did one set of downsampling where we just randomly selected the number of species from all Hall Arctic species to downsample to get the same kind of proportion of representation as like the tropical species had in our data set. And then we also did the converse, where we specifically targeted the cooperative breeding species in the tropical group, and downsampled that to from our like female song data set. Effectively, like if tropical species are more likely to be studied if they're cooperative, basically if they have this more interesting, behavioral paradigm, they may be more likely to have female song data as well. So we also downsampled the certain number of cooperative species that we had female song for to match the proportion of tropical species that were cooperative breeders, but we did not have female song for. So basically both of those ways to kind of use these, address these different like biases. And we found that the results were very similar. There kind of was always still a link between cooperative breeding and female song. Now, the direction of that link did sometimes change. There does seem to be evidence for bi-directional evolution there, but yeah, it was robust, and there's still a link in some direction.
Great, thank you. We're going to have to move on now. So yeah, we're on our last talk in the session. With Marc, would you like to share your slides?
Yes. Do you see my screen?
It's loading, yes. So we're looking forward to hearing about riding the wave. Take it away, Marc.
Okay. Hi, I'm Marc, and I'm going to, well, thanks for being here. I'm going to present part of my PhD thesis that is about invasion dynamics, morphological, and behavioral divergence in an invasive snake across an expanding invasion front. Invasive species dramatically affect the ecosystems they're introduced to. You have some examples of invasive species here on the left. And the impact of these introductions is even greater in island ecosystems because of their fragility of the ecosystems. But it also offers us the chance to observe and quantify the evolution in real time. Evolution not always comes with natural selection. There are other mechanisms that can also produce the evolution of traits. And one of these mechanisms is spatial sorting. Spatial sorting is the process through which dispersed individuals tend to accumulate in the invasion front of an expanding edge of an invasion. There, a sortative mating happens, and these dispersive traits get amplified generation after generation and are dispersed along the invasion range. So this process can lead, in the end, to shifts in behavior and morphology related to the system. The system I'm going to talk about offers us a great opportunity to explore spatial sorting in an ongoing biological relation. So the system we are working in is the island of Ibiza. It's an island located in the Mediterranean, and in recent decades, it has undergone rapid urban development that accidentally brought the protagonist of this story to the island. Around 2003, via olive tree trade, this species was introduced, and some previous studies already show how over 50% of the diet of Hemorrhoids lepis is based on Podarcis pituitensis, which is the other protagonist of this story. Podarcis pituitensis is the only native terrestrial vertebrate of the island, and because of the introduction of the snake, those populations that are in contact with the snake are becoming extinct. So what's our main question? Our main question is to answer if snakes from the fronts of invasion travel and explore more than those coming from the core of invasion. Our hypothesis is that yes, that this system reflects or matches what is found in other systems around the world, for example, the cane toads in Australia. And in order to do this, what we did was, first of all, to reconstruct the invasion history of the invasion. We combined different data sources in order to trace where the front of invasion was every year. And by doing this, we revealed that the invasion is accelerating year after year. We then captured some snakes along the island in order to have a representation of the whole gradient of invasion. And we built this experimental enclosure or arenas, where, as you can see in the bottom left GIF, a snake could freely explore for 1 hour while being recorded. We then quantified all these explorative behaviors, dividing between explorative behaviors painted in red in this plot and non-explorative behaviors painted in blue. in order to test for differences between the front and the core of invasion. Here you have a subsample of individuals. You can see how some individuals extort more than others. But if we look closer, if we look for a single individual, we can see that first there's a phase of them hiding from the arena, but then we can see when they engage visual exploration of the arena and when they engage facial exploration. And also all the behaviors they express during the 15 minutes trial, in this case, they are in the 15 minutes trial they are doing. So in order to test for differences in exploration and dispersal, what we did was analyzing the first 15 minutes of exploration of each snake after the emergence from the refuge. And what we did was split the 15 minutes in two groups. One that was the initial exploration that included the first 3 minutes of exploration of each snake, and then also the sustained exploration, which only accounted for the last 3 minutes of exploration during this 15 minutes trial. Having done this, I'm going to present the results. The first results we obtained were regarding when do snakes start exploring. We found that core of invasion snakes, both males and females, are more likely to start exploring when they come from the core than those coming from the front. These results might sound counterintuitive because at the beginning we were expecting that front of invasion would be more bolder to explore, but maybe what is happening is that those snakes that are standing, those snakes coming from the front, are facing always new habitats and risky environments that they have to be aware of. So being cautious might be favored during expansion, and there might be a spatial filter that selects for slow starters in the front of invasion. Regarding behavioral variation in their spatial exploration, we had some behavioral traits that we quantified, but we wanted to reduce their dimensionality. So we performed a PCA in order to see if there was an axis that explained behavioral, like spatial explorative behaviors versus non-spatial explorative behaviors. And we found that PC1 was useful to account for these differences in explorative behaviors. So when testing if there were differences in PC1 scores between core and front of invasion snakes, we found that core of invasion females had greater values of PC1 compared to front of invasion females. This means that sustained exploration drops in core of invasion females because we are testing only for the last 3 minutes of the trial, and that front of invasion females maintain exploratory behaviors for longer. Our next question was like, okay, we have these differences in PC1, but does these PC1 values translate to distance traveled, as the examples or graphic examples of trial sorting show? So when looking for differences in total distance traveled between the first 3 minutes of exploration and the last 3 minutes of exploration, we found that while there were no differences between distance traveled between core and front of invasion females, we found differences between core and front of invasion females regarding sustained exploration. So the exploration that happens after some time in the arenas. Core of invasion females traveled less distance during the last 3 minutes of exploration than they did during the first 3 minutes of the trial. But front of invasion females do the opposite. Front of invasion females show greater persistence in exploration compared to those coming from the core. We didn't find any difference in males. So for the moment, we've talked only about behavior, but what happens with morphology? Does morphology also change along the invasion gradient? As I told you in introduction, the function of this snake is leading to the collapse of lethal populations in the core area of the invasion. So it's leading also to to reduce food availability for those snakes coming from the core of invasion. So this might be driving a morphological change in the cranial morphology of these snakes coming from the core of invasion in order to adapt to alternative types of prey. In this case, small mammals, which are larger in size than lizards. So our hypothesis is that front of invasion snakes might retain head shapes suited for lizard hunting, while core of invasion snakes might present longer and broader jaws adapted to prey on larger types of animals. In this case, small mammals, as I said. So to do this, what we did was to X-ray a subsample of individuals. We could X-ray their heads. We identified some landmarks in their craniums that were repeatable between individuals and among individuals. And we measured the distances between these landmarks. And then once we have all these cranial morphology measurements, we tested for differences between core of invasion and front of invasion snakes. Although these results are preliminary, we are still working on this part of the thesis. The results point to the direction that core of invasion snakes have larger cranial elements than those snakes coming from the front of invasion. As I said, larger skulls or cranial elements in the core might be related to the bigger size of the prey they are predating on in the core of invasion, and revealing an adaptation for different alternative type of prey. So, as for the conclusions, we show that behavioral traits and spatial exploration shift along the invasion gradient. We also show that there might be a filter that selects for more cautious explorers towards the expanding edge of the invasion. And we also show that exploration is shaped by both sex and invasion area. And then the interaction between these two factors, sex and invasion area, is key to understand how the dynamics of the invasion progress. One of the main results is that females at the invasion front show sustained exploration over time. See a thing that we don't see or observe for the invasion females, and that this result is compatible with a special sorting process, although more experiments should be performed in order to test for this. Lastly, the invasion-driven ecosystem change that the introduction of snake has produced in the island of Ibiza might be influencing school morphology in the snakes coming from the core of invasion. So, having said that, thanks to all the team both in Ibiza and in Barcelona, and special thanks also to José and Eva Montes, without whom this wouldn't be possible without their collaboration.
Great, thank you for that talk. We have time for questions if anyone wants to post them in the Q&A or raise their hand. I'll get us started with a question. Do you have any data from the native range of these snakes to compare?
We're currently working on that because we know that we suspect that the snakes come from the peninsula, the Iberian Peninsula. So we're with people from Granada in the south of Spain in order to get some samples from there and see if there are differences both in morphology and genetics.
Great. Are there any other questions? All right. Well, I appreciate everyone sticking with us through some small technical difficulties. It's been a great group of talks. I really enjoyed this. There's a second behavior session later this afternoon, but I want to thank all our speakers again and thank everyone else for their participation. So thanks, everyone.
Thank you.